Guys, and welcome back to Excel Academy. So, like previously, we were talking about the translation, right? And we have discussed like how mRNA, tRNA, and ribosomes RNA are helping in the translation. Okay, so now in this session, we'll be starting with the initiation of translation. So, of course, we are going to talk first about the prokaryotic initiation. Once this is clear, then we'll move on to the eukaryotic. Okay, now for any translation to be successful, there are few events which must occur. First one is the ribosome should be recruited to the mRNA. Okay, so you have got the mRNA, right? And the ribosomes are needed for it to start. A charged tRNA must be placed into the P site of the ribosome. So guys, when you start translation, always remember, हम हमेशा क्या बोलते हैं that your new amino acyl or aminoacylated tRNA comes and sits on the A side. But in order to begin the translation, first it is going to sit on the P. Okay, then only A में आएगा and then it get transferred to the aminoacylated tRNA and so on. The last point is the ribosome must be precisely positioned over the start codon, right? So आपको यहाँ पे ध्यान देना है कि like all this reading frame and the RBS joining to the 30A subunit, it all are needed in order to start the translation. तो सबसे पहले क्या होगा आपका RBS the RBS is going to scan and get base paired with the 16S rRNA, right? So they are going to position in such a way that it is sitting on the your start codon. Okay. Now it has to be in such a way. The correct positioning of the ribosome over the start codon is critical because it is going to establish your reading frame for the translation. Okay. एक base भी shift हुआ आगे या पीछे, the entire ORF will change and eventually will result in the synthesis of wrong polypeptide chain. Okay. Now, so prokaryotic mRNA are initially recruited to the small subunit by base pairing to RNA. So this is what I was talking about. So you have got the RBS, right, just before your start codon, and ये RBS जो है आपका 16S RNA के साथ complementary base pair करेगा, ठीक है? Now once this is been assigned properly, okay, then your AUG, that is your start codon will come onto your P. तो इसका मतलब क्या? Your first tRNA, which is also known as initiated tRNA, is going to come and sit on the P side, correct? So this is your special tRNA, which is also known as the initiator tRNA. Okay, so initiated tRNA. So this is going to base pair with the start codon, usually AUG and GUG. So we know that AUG आपका methionine को कोड करता है, GUG आपका valine को कोड करता है. So as I mentioned, it has to start from somewhere, right? So evolution has adopted taking AUG as the start codon. There are very few organisms, like rare cases है जहाँ पे GUG are the start codon. Okay, so ऐसा नहीं है कि सिर्फ AUG ही रहेगा, but maximum species start codon is AUG. There, हाँ.
Ma'am?
Yes.
Your voice is like breaking and it's not clear, ma'am.
Okay, 1 second. Okay. So as I was saying, so special tRNA, initiated tRNA is there. Okay, ये जो है जो आपके सबसे पहले आपके codon सबसे पहले it is going to read your first codon. Okay. Now as I was saying, your start codon, either it is going to be your AUG, which is coding for methionine, or it is going to be GUG, which is going to code for the valine. So these are the very few rare cases जहाँ पे GUG is the start codon. Okay. Now AUG, GUG, they have a different meaning as they occur in the ORF, where they are read by the tRNAs. So tRNA is for methionine. Okay. So AUG is for your methionine and GUG is for your valine. Okay. Now, although initiated tRNA is your first charge with methionine, a formyl group is again added, okay, to the methionine amino group by a separate enzyme called MET-tRNA transformylase. Please, guys, this is important to remember. MET-tRNA transformylase. So, what does it mean? It means it is going to convert your methionine into formyl methionine. Now, the question is why does it do? कि ये methionine को formyl methionine क्यों करता है? So this is your assignment, today's assignment. Okay. सो क्वेश्चन क्या है यहाँ पे, क्यों वो मिथिनिन को फॉर्मल मिथिनिन करता है. सो वी नो द एंजाइम व्हिच डज, दैट इज योर मेट tRNA ट्रांसफॉर्माइलेज. बट व्हाई इट इज रिक्वायर्ड, व्हाई इट इज रिक्वायर्ड टू हैव अ formulated methionine in the case of the prokaryotes, and this is absent in the case of the eukaryotes. Okay, if you know the answer you can tell now. Do you know the answer, or I'll keep it as assignment and tomorrow we can discuss? Yes, Akshay, Shruti, right now do you have the answer?
No.
Okay, no problem, no problem. It's okay. So we'll discuss tomorrow. No problem. So I'll just repeat the question. So the question is, why does, in the case of prokaryotes, the first amino acid, which is methionine, gets formulated? Okay, simple. So we know the enzyme is met-tRNA transformase. So this is how it does. So this charged tRNA, initiated tRNA, is now known as F-met tRNA initiator फॉर्मल मिथ्यूनिन. कैन यू सी, दिस इज हाउ द नॉमेनक्लेचर इज बीन गिवन. हमने पहले कैसे डिस्कस किया था, इफ इट वाज वैलिन देन tRNA वैल, ऐसे दिखते हैं. ठीक है, सो व्हाट डज इट मीन, दिस मींस दिस tRNA हैज गॉट द एंटीकोडॉन फॉर वैलिन एंड नाउ इट इज चार्ज विद वैलिन एमिनो एसिड. ओके, अब यहाँ पे कैसे पढ़ेंगे इसको, F-met is the amino acid to which it is being charged. Okay, amino acid, this is your amino acid. tRNA I, why? Because this is your first initiated tRNA which is meant for F-met. Okay, so F-met matlab kuch nahi, just you have got the additional group over here, formal group is present. Now because N-formal methionine is a first amino acid, yes.
Suddenly I got an idea, but I don't know if it is correct or not. Ma'am, you have already told that the bacterial DNA is methylated. So to differentiate the DNA and its it is being converted into formyl. Formyl group is added to it.
Okay, first of all, okay, when I said DNA is methylated, I'm not getting the connection between the amino acid formulated and over there it is methylated. मुझे वो connection नहीं समझा. Why? हम यहाँ पे क्या डिस्कस कर रहे हैं, ट्रांसलेशन. What you said is a part of replication. Okay, I am not getting the connection between the two. Here it is amino acid, there it is nucleotides which is getting methylated. And here it is amino acid, only one, that is your first amino acid, only this amino acid is getting formal group attached.
Okay.
Huh? Okay, but it has to do something with distinguishing between the two. Okay, ये एक hint है. Okay, बीच में कुछ क्लिक होगा तो बताना. Anyways, good try. So because now N-formyl methionine is your first amino acid to be incorporated into polypeptide chain, one might think that all prokaryotic proteins have formal group at their amino termini, जबकि ऐसा नहीं होता है. ठीक है, as soon as your polypeptide chain comes out of the larger subunit, there are certain enzymes called deformylase. What they do, they are going to remove the formyl group. ओके और जरूरी नहीं है कि हर एमिनो एसिड में आपका मिथ्योनिन फर्स्ट एमिनो एसिड हो, बिकॉज समटाइम्स व्हाट हैपेंस इज दैट दे फॉर्म इनिशियल एमिनो एसिड्स. ओके, अगर सपोज लेट अस से दिस इज योर पॉलीपेप्टाइड चेन, ये आपका एन टर्मिनल हो गया, ये आपका सी टर्मिनल हो गया. ओके, नाउ इन फ्यू केसेस व्हाट हैपेंस इज दैट स्टार्टिंग ऑफ फ्यू एमिनो एसिड्स लाइक 10 to 12, ओके, दीस एमिनो एसिड्स दे एक्ट एज सिग्नल sometimes. Okay, signal matlab it is going to signal like to which part of the cell it is going to get recruited. क्या उसको साइटोप्लाज्म में रखना है या न्यूक्लियस में डालना है या माइटोकॉन्ड्रिया, क्लोरोप्लास्ट या आउट ऑफ द सेल. ओके, लाइक इट इज गोइंग टू बी अ सेक्रेटरी प्रोटीन. तो ये सिग्नल कैरी करता है. सो वंस द सिग्नल इज रिमूव, व्हाट हैपेंस देन द प्रोटीन इज गेटिंग its 3D structure. Okay, so it's not necessary that all the polypeptide chain which has started with AUG code, okay, start codon, तो methionine है आपका, It's not necessary that each polypeptide chain, this methionine is going to be of first amino acid. Clear? So there are many cases where the protein's initial part is getting cleaved. So that is also known as protein processing, where the additional amino acids which is not required in the structure, functional structure, it gets removed by the enzymes. So it's जरूरी नहीं है कि हमेशा आपका पॉलीपेप्टाइड या प्रोटीन जो है आपका फर्स्ट एमिनो एसिड आपका मिथ्योनिन होगा. Okay, now let's
Ma'am, does the formyl group help the ribosome identify from which part it's supposed to start for mRNA and then recognize as soon as it exits?
No. Then तो आपका ये फिर यूकैरियोट्स में भी प्रेजेंट होना चाहिए.
Yeah, but then prokaryotes में न्यूक्लियस के अंदर नहीं होता है ना सब कुछ. It happens like all over the cell. That's why I thought maybe that is the difference.
Okay, I'll give you another hint. The evolution has made the translation difference between the prokaryotes and eukaryotes for one reason. Okay, why? I'll give you one more hint that what if we get infected by these bacteria? And our body should understand that ये translation हमारा नहीं है.
Okay, so to distinguish between bacterial genetic material which is being passed through the
Okay,
cell.
so correct. So that's why, chalo, let us discuss now itself, no problem. So that's why, see, अब क्या है, let us say that we got infected by the bacteria, okay, prokaryotic species. So what will happen now? Even if the gene which gets integrated into the host genome sometimes, and they make sure that they take the host machinery for the translation, but I don't have fmet for my prokaryotic requirement. ठीक है, यूकैरियोट्स में नहीं होता है आपके पास fmet. So what will happen? Now I have stopped all the translation requirement of the prokaryotes. So it is helping the immune system, okay, of the eukaryotes in order to distinguish between the prokaryotic requirement of the translation from the eukaryotic requirement of the translation. ठीक है? So that's how the evolution has evolved. See, आपको हर एक चीज का benefit दिया है, but it has also made certain uniqueness so that in future किसी भी step में you can say that okay, this is prokaryotic, this is eukaryotic, and this should not get mixed. ठीक है? This is naturally, it has been evolved in the nature. Okay, जो भी अब हम आगे भी पढ़ेंगे natural selection and all. So this has had happened just because now we are humans, we are superior species. Okay, right now in the ecosystem. So we have got the intelligence, and that's why molecular cloning जो करके हम बोलते हैं. Here we are forcefully integrating the unwanted genes into the host genome, or taking the genes out of it for the benefit of the society, right? So this is something which we are doing against the nature, but we have got the why we are doing, because the simplest answer is to increase the lifestyle, or for the betterment of the society. This is what. Are we clear? Huh? So that's the reason why.
Formyl methionine have any significance in prokaryotes?
No. Like what I thought also in between starting, that it might be required, for the maybe protein folding or something like that, I thought initially. But if you see the structure, like crystallography structures, if you see, this formyl methionine has no direct contact with either the peptidyl transferase. Okay, there it has no no direct contact is there. Now even the tRNA, it is little different. Why? Because of it gets formulated. So you have got a specific enzyme to add to the methionine tRNA. Okay, it is your methionine tRNA. So this the formyl group is being added. ऐसा नहीं है कि पहले fmet होता है. Are you getting the point? So it's not that first it is fmet and then it goes to the tRNA. It is your first methionine tRNA to which the formyl group has been added by the enzyme. F-met transformylase. Okay. Now what we have seen is in the translation, the moment the polypeptide chain comes out of your larger subunit, okay, 50S subunit से आपका polypeptide chain बाहर आ रहा है, immediately this F-met group, okay, if it is required, see, this information is carried in the polypeptide chain only, whether that protein is going to get completely folded like this. If the entire protein is getting folded, then the formal group will be removed by the another enzyme called deformylase. अगर ठीक है, if that protein is meant to be sent to somewhere else, जैसे भी बैक्टीरिया में भी आप कहाँ भेज सकते हो प्रोटीन, either in the extracellular, right, it is going to be secretory, या फिर बैक्टीरिया में you have got different, ऐसे प्लाज्मा मेम्ब्रेन में भी आपके प्रोटीन्स रहते हैं, if you want to send it over there, so depending on the requirement of the protein recruitment, this F-met will be retained or it will be removed. ठीक है, so basically जब हम आगे पढ़ेंगे, like in the cell biology, you will see that all the proteins, okay, all the proteins, they carry a signal. Without this signal, the protein doesn't know where to go, to remain in the cytoplasm, to remain in the ER, Golgi, vacuoles, peroxisome, मुझे कहाँ जाना नहीं पता रहेगा. So always the signal is present. Now once the protein reaches its compartment, कि हाँ यहाँ पे मुझे काम करना है, then that signal gets cleaved. Okay, so we have got the enzyme called as a signal peptidases are there. Okay, so these signal peptidases will chop off that signal amount, and then आपका protein आपका fold हो जाता है and it performs its function. So chop off मतलब it gets degraded back to amino acids. ठीक है, so that's why I said many proteins, they are not going to have your first amino acid as methionine. Clear hai?
Yes.
So to distinguish, it helps the immune system to distinguish between the prokaryotic and eukaryotic translation machinery. This is the only answer I got, like why F-met is present in the nature. ठीक है, अगर आपको कहीं और कुछ और मिलता है, like if you're reading many research papers and all, please do share, like maybe those information will be later. तो हम ऐसे share कर सकते हैं if in case while reading, while going through, or maybe your teacher is telling something extra, then let me also know, because right now till now I think this is the only answer I have found and known in the text also. Okay, so now let's talk about the initiation part.
One more thing. About valine as a start codon, is it like protein specific or is it like species specific?
It is species.
Methionine, or so some species entirely use valine as a start codon.
Okay. So now about the initiation factors of translation. So from here onwards, guys, these are very important. ठीक है, कौन से factors हैं which is required in the initiation, which is required in the elongation and terminations. You need to remember their names as well as their function. ठीक है, and the order also in which they come. So each factor, it facilitates a key step in the initiation process. First one is your IF1, initiation factor 1. It prevents tRNA from binding to the portion of the small subunit that will become part of the A site. So what does it mean? So you have got the small subunit like this. ठीक है, so even they have got few regions, right, जहाँ पे आपका EPA रहेगा, तो ये IF1 क्या करेगा, यहाँ आके bind करेगा. Okay, so what will happen? So no other tRNA will come and bind over here. So अब क्या होना चाहिए next is your tRNA, initiated tRNA carrying the amino acid F-met should come and sit on the P side. ठीक है, so this is your initiated tRNA. Clear? This much is clear. So what is the first step? The IF1 is going to prevent binding of the tRNA to become a part of the A. Okay, now since IF1 is present, so what will happen? It is also preventing the binding of your 50S subunit. अब जहाँ already कुछ है आपके पास, तो क्या होगा? This 50S subunit is not able to come. ठीक है, तो इसके दो काम हो गए. हाँ.
Ma'am, can you repeat the sentence?
Yeah.
Yeah, it was.
Okay. I'll repeat. So I was saying IF1, it is going to prevent the tRNA binding to the portion where it is A. Okay. So if you have got the 30S subunit, so each subunit, they also have got these portions, little portions are there. Okay. Which is meant for E, P and A. Correct. Now IF1 will come and bind to this portion. This is your IF1. Okay. So what will happen now? No other tRNA can come. No other tRNA can come and bind to this portion. Okay. Only your initiator tRNA will come, which is having your F-formyl methionine. This is your initiator tRNA. Okay. Now, to this, because of this association, what will happen? The 50S subunit is not able to bind. Clear? It is not able to bind. Itna clear hai? Okay. Now, we have got another initiation factors. Okay, that is IF2, which is a GTPase. GTPase matlab kya? It is having GTP. IF2 is bound to your GTP. Again, if you remember in the previous lectures also, whenever such protein complexes are there, if gets hydrolyzed and it becomes GDP, IF2 loses its affinity for the initiation complex. Okay. Are you able to follow? So here, IF2, it goes and interacts with the small subunit, 30A subunit, IF1 and the charge initiated tRNA molecule. So how the association is going to be? This is your 30S. Okay. This is your first tRNA carrying the F-met. This is your IF1. Okay. Now here you have got your IF2 having GTP. Is this portion is clear?
Yes, ma'am.
Okay. Now by having this interaction, so IF2 along with the GTP is making sure the association of your first initiator tRNA and the 30S subunit, it is making sure that the entire initiation complex is getting ready. Okay. And it is, of course, going to prevent the other tRNAs from coming. Now, next what is coming is IF3. So it is going to bind to the subunit and blocks it from associating with the large subunit. Okay. Okay, I'm so sorry. Last time I said that it is IF1, right? It is IF3 only. IF3 is going to block your 50S subunit. Okay, so sorry about it. So because initiation requires a free small subunit, binding of IF3 is critical for a new cycle of translation. Okay. Now, once IF3 becomes associated with the small subunit, at the end of the previous round of translation, it helps to dissociate the 70S ribosome into its large and small subunits. So, इसका मतलब क्या? At the end of the translation, जब हम termination की बात करेंगे, there your IF3 will come again. So once this IF3 is coming again and binding to your smaller subunit, what will happen? The larger subunit will get separated. And one cycle of translation is over, and now these two are ready for another round of translation. Okay. So, okay. So now we have discussed about these three initiation factors. Let's go with the help of the diagrams as well. So if you see that, even though, I mean, we talked about IF3 at the end, but हमें वो सबसे पहले पढ़ना चाहिए था. So IF3 is responsible for binding to your E site so that it is preventing the association of your 50S subunit, ठीक है, to the 30 subunit. So this is your first step. Later on, your IF1 will come and bind to your A site, preventing any other charged tRNA to come. Correct? Then IF2 along with the GTP. IF2 along with the GTP is present. Now what will happen next? Your tRNA should come. So this IF2 along with the GTP is making sure that the complex, initiation complex is getting ready. Now, with all the three initiation factors bound, the small subunit is prepared to bind to the mRNA and the initiator tRNA. So guys, here the difference, okay? आपको initiation of जो translation का initiation है, वो बहुत अच्छे से ध्यान देना होगा because it is completely different for eukaryotes. ठीक है? तो अभी हमने क्या देखा? difference. Okay, not the difference, but what we have learned till now. What we have learned is that सबसे पहले आपका 30S subunit and the initiation factors are getting together. इसके बाद आपका mRNA आता है and tRNA आता है. Okay, whereas in the case of eukaryotes it is not like that. In the case of eukaryotes you have got the mRNA पहले. Okay. Now the binding of these two RNAs, that is either mRNA or tRNA, it is independent of each other. So ऐसा नहीं है that पहले mRNA जाके bind करेगा उसके बाद ही आपके पास tRNA आएगा. It is not like that. You can have your initiated tRNA coming first and then you can have your mRNA coming later. So it is independent of each other and can happen randomly. Okay. Now, as we know, so once mRNA is coming, it is going to base pair with your 16S rRNA of your 30S subunit. Okay. Now, meanwhile, the binding of F-met tRNA IF-met to the small subunit, it is facilitated by IF2 bound to GTP. Okay. So then what is going to happen is that the codon and the anticodon base pairing is going to be forming. Okay. Now, so once this base pairing is done, it makes sure that the position for the start codon at the P site is ready now. Guys, able to follow? So what we have discussed that 30S subunit में आपका पहले IF1, IF2, IF3 सब आते हैं, then only आपका mRNA आता है. एक बार mRNA आ जाएगा, it forms base pairing with the 16S rRNA. Then your tRNA is also coming. Now, which tRNA? F-met tRNA IF-met is coming, which goes and binds to the small subunit. क्या करेगा वो? With the help of the IF2 GTP, it is bound and making sure that the codon anticodon base pairing has been done. And it is ready कि वो अपना start position में present है. Okay, is this much clear, guys? Both of you?
Yes, ma'am. Yeah.
Cool. This is important to understand also. Okay. Now coming to the last step. So अब इसके बाद क्या आपका वो elongation होता रहेगा, ठीक है? Now for the last step, not sorry elongation. Now the 70S component आपका बनना चाहिए, correct? So अभी आपके पास सब कुछ है. You have got the initiator tRNA carrying the initiator amino acid, codon anticodon base pairing भी हो गया है. Everything is ready. Okay. Now what happens is that all this thing, it needs to result in the formation of complete intact ribosome. So यानी कि क्या इसका मतलब the 50S subunit, the largest subunit should come and get interacted with the 30S subunit. Okay. So when the start codon and the F-met tRNA IF-met base pairs, the small subunit undergoes change in conformation. Now, this conformation is going to result in the change in the conformation of IF3. That is your structural conformation. Okay. So what will happen? IF3 will lose its affinity for the 30S subunit. And now 30S is free of IF3. So IF3 nahi hai. अब आपका last subunit is free to come and bind to the small subunit which is carrying already IF1, IF2, mRNA and the initiator tRNA. Okay, so this is your initiation complex. Okay, look at the figure. So here we saw that initiation factor 3, 1, 2 and 2 is bound with the GTPs there. mRNA and tRNA is coming. So as I said before, the coming of mRNA and tRNA is irrespective of each other. Okay? Now what is happening? You can see that initiated tRNA is over here. Apna mRNA apni jagah pe aa gaya hai. Now what is happening? The moment initiated tRNA makes a codon anticodon over here at the start codon or the start position, there is a structural change onto the IF3. So IF3 gets released and then your 50S subunit comes and interacts. So this is your initiation complex ready. Okay. Yes. Now, few more points. In particular, this IF2, it acts as an initial docking site for the large subunit, and this interaction stimulates GTP's activity of IF2 GTP. Okay, so now we were talking, now we need to talk about the IF2 also, कि वहाँ पे IF2 GTP है वो किस काम के लिए, ठीक है? So it is actually helping the association of your 30s and 50s subunit का. So this association, okay, this is led by the activity of IF2 and bound GTP. So if it gets hydrolyzed, so we know that GDP, okay, once after hydrolysis, it has got reduced affinity. So what will happen? That time your IF2 is going to leave. Okay. Now this will also result in the removal of your IF1 also. क्यों? अभी हमारा initiation complex जो ribosome का है वो ready है. I don't want IF1. IF1 का क्या काम था? To block or to prevent the entry of any other charged tRNA, right? But now I don't want that blockage. So that blockage is removed with the help of the IF2 GTP hydrolysis. Okay. So the net result of initiation is the formation of 70S, जिसमें आपके पास है 50S, 30S. P site में आपके पास है initiated tRNA and a empty A site. Okay. Now they are ready to accept a charged tRNA and start the polypeptide synthesis. ओके आपको समझ में आया IF2 GTP का क्या फंक्शन है? समझा? हाँ, so what is happening till now? We saw that all this thing is coming. ठीक है, आपका IF3 when it was undergoing conformational change, because of that what will happen? IF3 is going to leave the 30S subunit. Now 50S subunit is coming and making association with the IF2. Okay, now once this association was happening, IF2 GTP will start getting, or it will activate its GTPase activity. यानी कि क्या? GTP is undergoing hydrolysis. This hydrolysis will result in the release of your IF2 as well as IF1. अब क्या है? IF2 की भी जरूरत नहीं है, IF1 की भी जरूरत नहीं है, वो सब चले जाएंगे. Now you have got initiated tRNA है, mRNA है, intact ribosome है, empty A site है. So what does it mean? It means it is ready for your polypeptide synthesis. अब आपके पास depending on the codon, that correct tRNA charged with the correct amino acid can be brought and the translation can be started. Okay. So here in this figure you can see IF3 गया, आपका last subunit आया. Okay, so this portion. Here you will see that IF2 and 1 is getting released after the hydrolysis of your GTP. So please do remember that IF2, the initiation factor 2, is also having the GTPase activity. Okay, and now your 70S initiation complex is ready. So it means your prokaryotic translation initiation has completed. अब इसके बाद क्या होगा? The elongations. Okay. Yeah, shall be stopping here now.